Good morning, class. My name's Silvana Iconi, and I'd like to welcome you to the first lecture of microbiology, Lecture 1, the history and scope of microbiology. So in this lecture, we're going to introduce the microbial world to provide a general idea of the organisms and agents that microbiologists study. Then we'll look at the historical development of the science of microbiology, then the scope, relevance and future of modern microbiology. The importance of microorganisms can't really be overemphasized. In terms of sheer numbers and mass, it's estimated that microorganisms contain 50% of the biological carbon and 90% of the biological nitrogen on Earth. And they greatly exceed every other group of organism on the planet. Also, they're found everywhere, from geothermal vents in the depths of the ocean to the coldest Arctic ice, to every person's skin, to every part of the plant. Microorganisms are major contributors to the functioning of the biosphere. They're indispensable for the cycling of nutrients that are essential for life. Microorganisms are also a major source of nutrients at the base of all ecological food chains and food webs. Most importantly, some carry out photosynthesis, and they rival plants in their capture of carbon dioxide and release of oxygen into the environment. Microorganisms that inhabit humans and other animals also play an important role. This includes helping the body digest food and producing vitamins such as vitamins B and K. Society, of course, also benefits from microbes, as they're necessary for the production of bread, cheese, alcohol, antibiotics, vaccines, vitamins, enzymes, and lots of other products. Now, although the majority of microorganisms play beneficial or benign roles, some are harmful and have disrupted societies over the millennia. Microbial diseases undoubtedly played a major role in historical events, such as the decline of the Roman Empire and the conquest of the New World. In 1347, the plague, or Black Death, an arthropod-borne disease, struck Europe with brutal force, killing 1/3 of the population, or about 25 million people, over 4 years. Over the next 80 years, the disease struck again and again, wiping out about 75% of the European population. Today, the struggle by microbiologists and others continues against killers like AIDS and malaria. Microbiology is often defined as the study of organisms and agents that are too small to be seen clearly by the unaided eye. That is the study of microorganisms. So because objects smaller than about 1 millimetre in diameter can't be seen clearly and have to be examined with a microscope, microbiology is primarily concerned with organisms and agents that are this small and smaller. However, there are some microbes, particularly some eukaryotic microbes, that are visible without microscopes. For example, bread molds, filamentous algae, and the very large bacteria such as Thiomargarita. So it's been suggested that other criteria should be used for defining the field of microbiology. For instance, an important characteristic of microbes is that they're relatively simple in their construction. They lack highly differentiated cells and distinct tissues. Another suggestion is that it should also be defined by its techniques. Microbiologists first isolate a specific microorganism from a population and then they culture it. Therefore, microbiology employs techniques such as sterilisation and the use of culture media that are essential for the successful isolation of microbes. Microorganisms are incredibly diverse and their classification has always been quite challenging. The early descriptions of plants or animals were just too simple. So, for example, some microorganisms are motile, like animals, some have cell walls, and some photosynthesize, like plants. Some are prokaryotic and some are eukaryotic. The word prokaryotes is derived from the Greek, meaning from the Greek pro and karyon, which means before nut or before kernel. Prokaryotes have a much simpler morphology than eukaryotic cells and they lack a true membrane delimited nucleus. In contrast, eukaryotic cells have a membrane enclosed nucleus and they're more complex morphologically and are usually larger than prokaryotic cells. These observations led to the development of a classification scheme that divided all organisms into 5 kingdoms: the Monera, Protista, Fungi, Animalia, and Plantae. Microorganisms are placed into the first 3 of these kingdoms, except for the viruses, which are acellular and have their own classification system. But we'll come back to the viruses in another lecture. The comparison of ribosomal RNA, begun by Carl Woese in the 1970s, was actually quite instrumental in demonstrating that there are in fact 2 very different groups of prokaryotes, bacteria and archaea, which are classified together in this 5-kingdom system. Taxonomists now believe that the 5-kingdom system is too simple, but although a few alternatives have been suggested, most microbiologists believe that organisms should be divided into 3 domains. Bacteria, which includes the true bacteria, otherwise known as eubacteria, archaea, and eukarya. And of course, eukarya includes all the eukaryotic organisms. Note that the word bacteria is plural, singular, bacterium. Archaea is plural, singular is archaeon. Bacteria are prokaryotes that are usually single-celled. Most have cell walls that contain the structural molecule called peptidoglycan. Bacteria are abundant in soil, water, the air, and they're major inhabitants of the skin, mouth, and intestines of animals. Some live in environments that have extreme temperatures, pH, or salinity. Some bacteria cause disease, but most are non-pathogenic and play more beneficial roles in the environment, such as cycling elements in the biosphere, breaking down dead plant and animal material, and producing vitamins. One group of bacteria called the cyanobacteria produce significant amounts of oxygen through the process of photosynthesis. Archaea are also prokaryotes and they're distinguished from bacteria by many features, most notably their unique ribosomal RNA sequences. Archaea lack peptidoglycan in their cell walls and they have unique membrane lipids. Some of the archaeans have unusual metabolic characteristics, such as the methanogens, which generate methane gas. Many of the archaeans are found in extreme environments. Pathogenic archaea have not yet been identified. The domain eukarya includes microorganisms classified as eukaryotic. That is, the protists, the group known as the protists, which includes unicellular algae, protozoa, slime molds, water molds, and fungi. The algae are photosynthetic protists that, together with the cyanobacteria, produce about 75% of the planet's oxygen. Algae are also the foundation of aquatic food chains. The protozoa are unicellular, animal-like protists that are usually motile. Many of the protozoa function as the principal hunters and grazers of the microbial world. They obtain nutrients by ingesting organic matter and other microbes, including bacteria. Protozoa are found in many different environments, and some of them are even normal inhabitants of the intestinal tracts of animals, where they may aid in the digestion of complex materials such as cellulose. And of course, a few of the protozoa cause disease. Slime molds are protists that are like protozoa in one stage of their life cycle, but they're like fungi in another part of their life cycle. In the protozoan phase, they hunt for and engulf food particles, consuming decaying vegetation and other microbes. Water molds, as their name implies, are found in the surface water of freshwater sources and also in moist soil. Water molds feed on decaying vegetation such as logs and mulch. Some water molds have caused devastating plant infections, such as the Great Potato Famine of 1846 to 1847. The fungi includes a diverse group of microorganisms that range from unicellular forms, that is the yeasts, to molds and mushrooms. Molds and mushrooms are multicellular fungi that form thin thread-like structures called hyphae. And these hyphae absorb nutrients from their environment, including the organic molecules that they use as a source of carbon and energy. The viruses are regarded as acellular entities, and they must invade a host cell in order to replicate. They're the smallest of all the microbes. The smallest is about 10,000 times smaller than a typical bacterium, but their size belies their power. They cause many animal and plant diseases, and they've caused epidemics that have shaped human history. Diseases such as smallpox, rabies, influenza, AIDS, the common cold, and some cancers are caused by viruses. Even before microorganisms were first seen, some investigators suspected their existence and also suspected that microorganisms were responsible for causing disease. Among others, the Roman philosopher called Lucretius, about 55 BC, suggested that disease was caused by invisible living creatures. But the earliest microscopic observations appear to have been made between 1625 to 1630 on bees and weevils by the Italian Stuluti. And he used a microscope that was probably supplied by Galileo. But the first person to publish extensive, accurate observations was the amateur microscopist, pictured here, Antony van Leeuwenhoek of the Netherlands. Van Leeuwenhoek was a draper and haberdasher, but he spent much of his time constructing simple microscopes. On the left here we can see a brass replica of Antony's microscope, which could magnify around 50 to 300 times. The inset photo shows how one of these microscopes is held. Beginning in about 1673, Antony started sending detailed letters describing his findings to the Royal Society of London. And it's clear from one of his drawings here on the right that he was seeing both bacteria and protozoa. As important as Antony's discoveries were, the development of microbiology languished for the next 200 years. Very little progress was made, primarily because microscopic observations don't provide sufficient information to understand their biology. So for this discipline to develop, techniques for isolating and culturing microbes in the lab were needed. And these techniques began to be developed as scientists grappled with this old conflict over spontaneous generation. From the earliest times, people believed that living organisms could develop from non-living matter, that is, spontaneous generation. Even Aristotle in 384 to 322 BC thought this. This view was finally challenged by the Italian physician Francesco Redi in the 1600s, who carried out a series of experiments on decaying meat and its ability to produce maggots spontaneously. Redi placed meat in 3 containers, which you can see in this image on the slide. One container was uncovered, the second was sealed with paper, and the third was covered with a fine gauze that would exclude flies. Flies laid their eggs on the uncovered meat and maggots appeared. The others didn't produce maggots spontaneously, however, the flies were attracted to the gauze and they laid their eggs there and maggots appeared from the eggs. Thus, maggots did not spontaneously appear as previously believed. And similar experiments by others helped to discredit the theory for other organisms. Yet, even with this experiment, this classic experiment, some people still proposed that microorganisms still arose by spontaneous generation, even if larger organisms like maggots did not. And they pointed out that boiled extracts of hay or meat would give rise to microbes after sitting for a while. So could spontaneous generation be true still for microorganisms? In 1748, the English priest John Needham reported the results of his experiment. He boiled mutton broth in flasks, sealed the flasks after boiling. They became contaminated. But he misinterpreted these results. He thought the organic matter contained a vital force that could confer the properties of life on non-living matter. A few years later, Spallanzani, the Italian priest and naturalist, improved on Needham's experimental design by first sealing glass flasks with water and seeds, then boiling for 3/4 of an hour. There was no contaminating growth as long as the flasks remained sealed. He proposed that air carries germs to the culture medium. But the supporters of spontaneous generation said that heating the air had destroyed its ability to support life. Enter the great French chemist and microbiologist Louis Pasteur. He was provoked to settle the matter once and for all. First, he filtered air through cotton and found that objects resembling plant spores had been trapped. If a piece of cotton was placed in sterile medium after air was filtered through it, microbial growth occurred. Next, he placed nutrient solutions in flasks, heated the necks of those flasks in a flame, and then drew out the neck in a variety of curves while keeping the ends open to the atmosphere. He then boiled the solutions for a few minutes and allowed them to cool. The result, no growth took place even though the contents of the flasks were exposed to the air. He said that dust and germs would be trapped on the inner walls of the curved necks. If the neck was broken, then growth commenced immediately. So Louis Pasteur had not only resolved the controversy by 1861, but he had also shown how to keep solutions sterile. Here are some of the flasks used by Louis Pasteur to disprove the theory of spontaneous generation. John Tyndall dealt the final blow to spontaneous generation in 1877 when he demonstrated that dust carries microorganisms. He showed that if dust was absent, then nutrient broth would remain sterile even if directly exposed to the air. The role of microorganisms in causing disease was still not immediately obvious. Establishing this connection depended on the development of techniques used for studying microbes. Most people believed that disease was due to causes such as supernatural forces or poisonous vapours called miasmas, or imbalances among the four humours thought to be present in the body and shown here on this slide. The role of the four humours, blood, phlegm, yellow bile and black bile in disease, had been widely accepted since the time of the Greek physician called Galen. Louis Pasteur's work with the swan neck flasks ushered in the golden age of microbiology between 1857 to 1914. Within 60 years, a number of disease-producing microbes were discovered, and great strides were made in understanding microbial metabolism and techniques for isolating and culturing microorganisms were improved. Support for the idea that microbes cause disease, that is the germ theory of disease, began to accumulate. Agostini Bassi was one of the first. He showed in 1835 that a silkworm disease was caused by a fungus. And he also suggested that many diseases were caused by microorganisms. More evidence was provided by the plant pathologist M.J. Berkeley, who showed that the great potato blight of Ireland was caused by a water mould. De Barry in 1853 showed that smut and rust fungi caused cereal crop diseases. Following his successes with the study of fermentation, Louis Pasteur was asked by the French government to investigate a disease of silkworms that was disrupting the silk industry. And after several years of work, Louis Pasteur showed that the disease was caused by a protozoan parasite. The disease was then controlled by raising caterpillars from eggs produced by healthy moths. The English surgeon Joseph Lister provided indirect evidence that microorganisms were the causal agents of disease. Lister was impressed with Louis Pasteur's studies on the involvement of microorganisms in fermentation and in putrefaction. And based on these studies, Lister developed a system of antiseptic surgery designed to prevent microbes from entering wounds, as well as methods for treating instruments and surgical dressings. So instruments were heat sterilised and phenol was used on surgical dressings and at times sprayed over the surgical dressing as shown here. This approach by Lister was incredibly successful and transformed surgery after Lister published his findings in 1867. And it also provided very strong indirect evidence for the role of microbes in disease, because phenol, which was discovered in coal tar only a few years earlier and referred to as carbolic acid, kills bacteria, and this also prevented wound infections. Now just note that surgeons didn't wash their hands in those days and didn't even change their gowns in between surgeries. They would be proud of the bloodstains on their gowns and wear them repeatedly as a sign of experience and refer to the good old surgical stink. The final proof came in the direct demonstration that a particular microbe causes a specific disease. The German physician Robert Koch was studying the disease anthrax by using a set of criteria developed by his teacher, Jacob Henle. And what Koch did was he injected healthy mice with material from diseased animals. The mice became ill, and then after doing this through a series of 20 mice, he incubated a piece of spleen containing the anthrax bacterium in beef serum. The bacteria grew, they reproduced, and they produced spores called endospores. Then when the isolated bacteria or their endospores were injected into mice, the disease called anthrax developed. And these criteria that Robert Koch used are now known as Koch's postulates, and they're still used today, although sometimes in a modified way, to establish the link between a particular microorganism and a disease. So here we have Koch's 4 postulates. The first postulate states that the microorganism must be present in every case of the disease, but absent from healthy individuals. The second postulate is that the suspected microorganism must be isolated from the diseased individual and grown in a pure culture. In other words, it's grown in the lab using nutrient media. The same disease must result when that isolated microorganism is inoculated into a healthy host, and the same microorganism must be isolated again from that diseased host. Now keep in mind that it is difficult to always satisfy these postulates. There are sometimes exceptions to these criteria. So for example, if we look at that first postulate, which says that the microorganism must be present in every case of the disease, but absent from healthy individuals. It's quite possible that that same microorganism is present in a healthy individual, but doesn't cause any manifestation of the disease due to that individual's immune system. Let's look at that second postulate. The suspected microorganism must be isolated and grown in a pure culture. Well, it's not always possible to grow microorganisms in the lab, and this is due to a lack of knowledge regarding the biology and the nutritional requirements of that microorganism. Here we can see how Robert Koch applied these postulates to demonstrate that the bacterium called Mycobacterium tuberculosis was the causative agent of tuberculosis. During Koch's studies, it became necessary to isolate suspected bacterial pathogens in pure culture. That is, a culture that contains only one type or one species of microbe. At first, Koch cultured bacteria on the sterile surfaces of cut and boiled potatoes, but this was unsatisfactory because the bacteria wouldn't always grow well. Eventually, he developed culture media that we'll talk about in another lecture, using meat extracts and protein digests because of their similarity to body fluids. He first tried to solidify the media by adding gelatin, and although this was okay, it wasn't ideal because many bacteria can digest gelatin, and also it melts at temperatures above 28 degrees. A better alternative was actually suggested by Koch's assistant's wife, who was using a substance called agar as a solidifying agent to make jellies. Now, agar, which was discovered by the Japanese from the cell walls of certain red algae, is not digested by most bacteria, and it doesn't melt until it reaches a temperature of 100 degrees. Also, once it did melt, it didn't actually solidify until it cooled down to about 50 degrees. And this eliminated the need to handle boiling liquid, and it provided time to manipulate the medium. So, for example, it could be handled with ease to pour agar plates and add antibiotics and other amendments without destroying them with high temperatures. So this nutrient broth and nutrient agar that were developed are still widely used today, and we'll also be using them in your prac classes. Another important tool that was developed in Koch's lab was the Petri dish, which is obviously a container for holding solidified media. And it was named after Richard Petri, who devised it. Other developments during this time involved the discovery and study of viral pathogens. The discovery of viruses was made possible when one of Louis Pasteur's associates, Charles Chamberlain, constructed a porcelain bacterial filter in 1884. And you can see the filter pictured here. Two scientists by the name of Ivanovsky and Beyerink used this porcelain filter to study a disease of tobacco called tobacco mosaic disease. And what they found was that plant extracts and sap from diseased plants were still infectious even when filtered with Chamberlain's filter. So because the infectious agent passed through a filter that was designed to trap bacterial cells, that told them that the agent must be something smaller than a bacterium. This led Beyerink to propose that the agent was a filterable virus, virus coming from the Latin meaning poison. And eventually, viruses were shown to be small, acellular, infectious agents. During this period, progress was also made in determining how animals resisted disease, and also in developing techniques for protecting humans and livestock against pathogens. An English doctor by the name of Edward Jenner had heard a milkmaid comment that she couldn't catch the dreaded smallpox disease, one of the biggest killers of the day. And she couldn't catch this disease because she had once contracted cowpox. Now, cowpox is caused by a virus that's very closely related to the smallpox virus. And cowpox can be transmitted from cows to humans, but it only causes a mild, short illness. So what Jenner did was he cut the arm of a young boy called James and infected the wound with blood lymph from a cowpox lesion. Later on, he infected the boy with smallpox, but the boy was resistant to the disease. So without even understanding the molecular basis of what he'd done, Jenner had developed the vaccination procedure. Louis Pasteur and his colleagues also studied a disease called chicken cholera. and they discovered that incubating their bacterial cultures for a long period of time before transferring to fresh culture would attenuate the bacteria, meaning that the bacteria would lose their ability to cause disease. Then if the chickens were injected with this attenuated bacteria, they remained healthy but became resistant to the disease. Pasteur called the attenuated culture a vaccine, derived from the Latin word vacca, which means cow, and this was in honour of Edward Jenner. Louis Pasteur prepared also a rabies vaccine using a very different approach. The pathogen was attenuated by growing it in an abnormal host, a rabbit, and then after death, the brains and spinal cords of the rabbits were removed and dried. While he was performing these studies, a 9-year-old boy was bitten by a rabid dog and brought to Pasteur. The boy's death was a certainty, so Pasteur agreed to try the treatment. The boy was injected 13 times over the next 10 days with increasingly virulent preparations of the attenuated virus. He survived. Although humans had been using microbes for thousands of years unknowingly, industrial microbiology developed in the most part due to the work of Louis Pasteur and others on the alcoholic fermentations that yielded wine and also other alcoholic beverages. Now, it was widely believed by the leading chemists at the time that fermentation was due to a chemical instability that degraded the sugars to alcohol. Louis Pasteur did not agree with this. He actually believed that fermentation was due to the action of microorganisms. In 1856, an industrialist in France requested Louis Pasteur's assistance. His business produced ethanol from fermentation of sugar beets, but recently the alcohol yields had declined and the product had become sour. Pasteur discovered that the fermentation was failing because the yeast normally responsible for alcohol fermentation had been replaced by microbes that produced lactic acid rather than ethanol. So in demonstrating this, Pasteur then showed that all fermentations were due to the activities of specific yeasts and bacteria, and he published several papers on fermentation. Pasteur's studies also led to a study of wine diseases and the development of pasteurisation to preserve wine during storage. The field of microbial ecology developed when a few of the early microbiologists decided to study the role of microbes in the environment. In particular, the carbon, nitrogen and sulfur cycles taking place in soil and aquatic habitats. The Russian microbiologist Winogradsky made lots of contributions to soil microbiology. He discovered many interesting metabolic processes such as nitrogen fixation, the decomposition of cellulose, and also that many bacteria could incorporate carbon dioxide just like photosynthetic organisms do. Beyerink was also a great general microbiologist. He isolated the nitrogen-fixing Azotobacter and also the root nodule bacterium called Rhizobium that also fixes nitrogen. He also isolated sulfate-reducing bacteria. So as one scientist writer emphasised, we live in the age of bacteria. They were the first living organisms on the planet. They live everywhere that life is possible. They're more numerous than any other kind of organism. The global ecosystem depends on their activities, and they influence human society in many ways. Because microbiology is so diverse, today's modern microbiology is a very large discipline with lots of different specialties. Microbiology has both basic and applied aspects. Basic aspects are concerned mostly with the biology of individual groups of microorganisms and includes studies in the areas of microbial physiology, genetics, molecular biology and taxonomy, where applied aspects are concerned with practical problems relating to disease, water, food and industrial microbiology. But it's important to note that the basic and applied aspects are often intertwined. Basic research is often conducted in applied fields and applications usually arise out of basic research. Here are just some examples of the specialties found within the field of microbiology. So medical microbiology is concerned with diseases. Immunology is concerned with how the immune system protects a host from pathogens. Microbial ecology is concerned with the relationship that organisms have with their environment. Microbial genetics and molecular biology are concerned with understanding how genetic information functions and regulates the development of cells and organisms. The future of microbiology is quite clear. Firstly, because microbiology has a clearer mission than most other disciplines. And second, microbiology has enormous practical significance. Microbiology is required to combat current and emerging and re-emerging diseases. It's required to develop industrial technologies that are more environmentally friendly and more efficient. It's required to combat the spread of multiple antibiotic resistance and chemical resistance. Microbiologists are being called on to create new drugs and vaccines to further our understanding of host defences, not only in animals but also in plants. We also have this continuing need to protect food and crops from microbial damage. And we'll need to use techniques in molecular biology and recombinant DNA technology to solve many of these problems. Understanding microbial diversity and ecology is actually one area that needs an enormous amount of work done. It's estimated that less than 1% of Earth's microbial population has been cultured. I'd like to leave you with The Future of Microbiology by the microbiologist René Dubos.